Hello students. Today in this session we are going to study about fertilization and implantation. This is a topic of third chapter of class 12th biology, which is human reproduction. So let us begin with our class. Before we start the topic fertilization, let us study about the structure of an ovum. The human egg is very small, only about 0.115 millimeter in diameter. It is non-motile. The yolk is negligible because it gets implanted within about 7 to 8 days after fertilization in the uterus and draws the nourishment from the mother through the placenta. The human egg or ova at ovulation is at the secondary oocyte stage. Now let us study the structure of an ovum. The ovum contains a haploid nucleus and a large amount of cytoplasm. The ova is surrounded by radiating rows of follicle cells called corona radiata, forming a dense crown. Corona radiata protects the ova till fertilization and gets broken down only by acrosomal enzymes released by the sperm. Inner to corona radiata is a transparent clear zone called zona pellucida secreted by the corona cells. It undergoes significant changes and thickens after fertilization to stop the entry of other sperms. Next to zona pellucida on the inner side is the perivitelline space which block polyspermy. So having understood about the structure of an ovum, let us now discuss about fertilization. So what is fertilization? The process of fusion of a sperm with an ovum is called fertilization. So like we saw, fertilization is the process of fusion of sperm with an ovum. So during copulation, semen is released by the penis into the vagina. The motile sperms swim rapidly through the cervix, enter into the uterus and reach the ampullary isthmic junction of the oviduct. A sperm comes in contact with the zona pellucida layer of the ovum and induces changes in the membrane to block the entry of additional sperms. Thus, it ensures that only one sperm can fertilize an ovum. The enzymes of the acrosome of sperm help to dissolve zona pellucida and plasma membrane of the ovum, and sperm head is allowed to enter into the cytoplasm of the ovum. As the sperm enters the ovum, which is actually a secondary oocyte, it gets activated and undergoes the second meiotic division. As a result of this, the secondary oocyte produces a haploid ovum and a polar body. The polar body ultimately degenerates and the ovum contains haploid number of chromosomes. The fusion of the sperm and egg restores the diploid number of chromosomes. The single diploid cell formed is called zygote. It contains both the paternal and the maternal chromosomes. It is the beginning cell of the new multicellular individual. Immediately after fertilization, the zygote undergoes the first mitotic division. Now let us study how the sex of the child is decided. The sex of a child is decided as zygote is formed. It is determined by the chance fusion of an egg and a sperm. Since females are XX as a result of meiosis during the formation of gametes, all the eggs contain a single X chromosome. Since males are XY, as a result of meiosis, they produce two kinds of sperms. 50% of sperms contain X chromosome and 50% contain Y chromosome. Now, if the sperm containing an X chromosome fertilizes an egg, the zygote is XX and develops into a female. If the sperm containing a Y chromosome fertilizes an egg, the zygote is XY and develops into a male. Since there is an equal probability of an egg combining with X containing sperm or Y containing sperm, there is 50% chance of an offspring being a female and 50% chance of being a male. Scientifically, it can be said that the sex of the child is determined by the father and not by the mother. Now, let us look at what is implantation. The attachment of the embryo to the thickened inner wall of the uterus 7 to 8 days after fertilization is called implantation. Now let us study about the development of zygote into an embryo and its implantation. Zygote divides rapidly by mitotic division as it moves through isthmus of oviduct towards uterus. This is called cleavage. As a result, 2, 4, 8, 16 daughter cells are produced which are termed as blastomeres. Embryo with 8 to 16 blastomeres is called a morula. Morula means little mulberry. The morula continues to divide and transforms into a large mass of cells called a blastocyst, which passes further into the uterus. Blastomeres in the blastocyst are arranged into an outer layer called a trophoblast and an inner group of cells attached to trophoblast called a inner cell mass. The trophoblast layer gets attached to the cells of the endometrium and the inner cell mass gives rise to the embryo. After attachment, the cells of the endometrium divide rapidly and cover the blastocyst. So, the blastocyst gets embedded in the endometrium of the uterus. This is called implantation, which leads to pregnancy. The given figure shows how the primary follicle changes into secondary follicle by the addition of more layers of granulosa. Secondary follicle is transforming into tertiary follicle. Within the tertiary follicle, the primary oocyte grows in size and completes its meiotic division to produce two haploid cells, the secondary oocyte and the polar body. We have already discussed all these events in the previous class. The secondary oocyte undergoes the second meiotic division. As a result, an ovum is produced. At the same time, the tertiary follicle further changes into the Graafian follicle. The Graafian follicle now ruptures and the secondary oocyte is released from the ovary by the process called ovulation. The infundibulum of fallopian tube collects this ovum after ovulation. Sperm fuses with the egg and fertilization takes place in the fallopian tube. As the zygote moves through the isthmus of the oviduct, the zygote undergoes repeated mitotic division called the cleavage division. The first cleavage division divides the zygote into two blastomeres. The second cleavage division divides both the blastomeres, resulting in 4-cell stage. Third cleavage divides all the 4 blastomeres into an 8-cell stage. The embryo with 8 to 16 blastomeres is called a morula. The morula continues to divide and transforms into blastocyst as it moves further into the uterus. The blastomeres in the blastocyst are arranged into an outer layer called a trophoblast and an inner layer called inner cell mass. The trophoblast layer then gets attached to the endometrium. As a result, implantation of the blastocyst in the uterus occurs. At the same time, inner cell mass of blastomeres gets differentiated as the embryo. After attachment, the uterine cells divide rapidly and covers the blastocyst. As a result, the blastocyst becomes embedded in the endometrium of the uterus. This is called implantation and it leads to pregnancy. Thank you.